Hi, I'm Tatiksha. Biochemistry is crucial in understanding the body's basic functions at a molecular level, such as digestion, energy derivation, metabolism, gene function, and immunological resistance. Medical biochemistry is essential for research in medical science branches.
Proteins are large-sized heteropolymeric macromolecules, having one or more polypeptides. A single polypeptide must be at least 50 amino acids long in order to be called as proteins. In a cell, protein is synthesized in nucleus and cytoplasm. Firstly, DNA within the nucleus is transcribed to form mRNA. This mRNA then leaves nucleus so as to enter cytoplasm. Here, ribosomes are attached with mRNA, and finally genetic information of mRNA is translated as proteins. This newly formed proteins are variously folded as per biological requirement. This process is called central dogma of molecular biology. Protein folding leads to different level of structures in a protein. We shall now briefly discuss about levels of structure in a protein. Primary level. It describes fundamental structure of a protein. This includes number of polypeptides, number and sequence of amino acids in each polypeptide. Left end of a polypeptide chain is called N-terminal, whereas right end is called C-terminal. Secondary level. It is of three types. Alpha helix, it is right-handed spiral structure. For example, keratin, myosin, tropomyosin, fibrin, etc. Beta pleated, two or more sheets are interconnected by hydrogen bonds. For example, parallel beta sheet is represented by beta keratin, whereas antiparallel beta sheet is shown by fibrin of silk. Collagen helix, most abundant protein in our body. Usually, three polypeptides are coiled around one another. Proline and hydroxyproline gives locking effect, thus stabilizing the structure. Tertiary level. In this level of structure, there is bending and folding of various types to form spheres, rods, or fibers. Active sites or polar side chains of the proteins are often brought towards the surface. Certain other side chains, for example, hydrophobic, are brought to the interior of protein. A protein often requires another protein called chaperonin for development of its tertiary structure. Quaternary level. It is found only in multimeric proteins. Each polypeptide develops its own tertiary structure and functions as subunit of protein. For example, hemoglobin is made up of 2 alpha and 2 beta polypeptide chains. Functions of proteins. Frederick Sanger worked out on sequence of amino acids in the polypeptide chains of hormone insulin. Manelin, a protein from African berry, has intensely sweet taste and is beneficial for diabetic patients. Change in sequence of amino acids in a particular protein can cause serious disease. For instance, if in hemoglobin chain, 1-glutamic acid is replaced by valine, it can cause sickle cell anemia. Streptokinase is produced by bacterium Streptococcus and is used clinically to remove blood clots from blood vessels and is known as clot buster. Thank you.